So good morning, everyone, and thank you, Professor Abbott, for that introduction. Thanks to all of the organizers for making this possible. And so as Professor Abbott mentioned, today I'm going to be giving a talk on how can we design surfaces and interfaces to design anti-fouling and antimicrobial systems. Our research group, it's broadly interested in many of these topics. But today I'm going to show how we can create a polymer matrix in which we can infuse an oil, creating an interface to which bacteria cannot adhere or attach to. So I think everyone here gave a great introduction on why it's important to study bacteria and how can we manipulate them. So I'm going to get started with the protagonist of my story for creating these polymer matrices. So the protagonist will be the polymer polyvinyl dimethylacetone, or PBDMA. This polymer is unique in the sense that it's highly reactive. So this polymer, the ring is a 5-member ring, so it has a high strain, and also it's very electrophilic. So that allows this polymer to react in click-type chemistry reactions with nucleophiles such as primary amines. Click-type reactions are just reactions that occur very rapidly and do not form any byproducts. So these allow us to, this polymer has a very versatile chemistry, allowing us to design many polymers with different side chain functionalities. But what I'm going to talk today is how can we use this polymer for the template or synthesis of many thin coatings and films. So to do that, we use the layer-by-layer assembly process, which many of you may know from the work on multi-layers of polyelectrolytes, where we have used the same technique to develop multi-layer films which are covalently cross-linked, and we call the reactive layer-by-layer assembly. So the basic idea of this process is that we start with a substrate which we dip on a solution of this polymer, and the absorption of this polymer to the interface, it's absorbed to the interface. Then by adding a second layer of the other polymer, we can then continue that process and create a multi-layer film that it's covalently cross-linked with the formation of 2 amide bonds. If we compare these to conventional polyelectrolytes, these films are, since they are covalently cross-linked, they're highly stable at different conditions, such as pH extremes or high ionic strength. However, we have seen that these films have unique properties because some of those acetone side chains do not react. Some of them are still unreacted after the film assembly. So that allows us to modify these surfaces after fabrication for controlling the surface properties, but also the bulk properties of these multi-layers. So we have used that approach to design many different thin films and coatings for an array of different applications. And for the purpose of this presentation today, I'm going to talk about how can we use those coatings to create anti-wetting and anti-fouling surfaces. So the work started with previous observations of members of my research group, where they saw that depending on the functionalization that you will give these films, you can obtain different interfacial properties. For example, if we reacted the residual acetones with hydrophobic primary amines, such as acylamine, we will get surfaces that are hydrophobic or have a very high contact angle. However, if we functionalize these films with molecules that were more hydrophilic, then we could create surfaces that were more hydrophilic. And then we began thinking about how can we further. We know that we can control the surface chemistry of our materials just by reacting with different molecules, but we also can change the topography features of these surfaces by the conditions at which we fabricated these films. So that's when we began to think, how can we change the surfaces from the hydrophobic realm to the superhydrophobic realm? And many of you may be familiar with the lotus leaf, which is a leaf that is very superhydrophobic, meaning that it's extremely non-wetting or exhibits contact angles that are greater than 150. So by controlling the conditions at which we fabricated these films, we were able to create more porous multi-layers, like the one I'm showing here. I'm going to talk more about that in a minute. But that is useful for a range of different applications that we have demonstrated. But what I'm going to talk today specifically, it's how can we use these films and what makes the films that we made unique in the sense that they are reactive and that they are very porous. So if we look, this is a top-down view of an SEM image of one of these multi-layer films fabricated using PBDMA and BPI. This film is around 80 micrometers thick, and we can observe that after depositing around 35 bilayers, we see that a lot of nano fissures as well as micro fissures. That is the features that make this possible, the films to become super hydrophobic. And since these films are very thick, if we were going to scratch the surface of these films, we will see that these same features that we see at the surface propagate along the bulk. So we can start thinking that our material has a very high surface area and that we can use that to incorporate other interfaces or molecules into our films. And that's exactly what has been demonstrated with slippery liquid infused porous surfaces, or SLIPS, a technology that was developed at Harvard by Professor Johan Eisenberg. And what we have, the basic principle is that if we start with our material, a polymer, a hydrophilic polymer matrix, and we incorporate and infuse an oil phase into that polymer, we can create a defect-free surface. And the oils that we can use to synthesize these materials can range from perfluorinated oils to silicon oil or any other type of oil such as olive oil or coconut oil, for example. And what this does is that creates an interface that every other liquid or material that is incompatible with our surface, it will just slide off. And by researchers in MIT, it has been used to develop consumer products in which the coating of ketchup bottles allows to release all the ketchup that is inside the bottle, minimizing the amount of ketchup that is wasted. But we're more interested in how can we use these surfaces to prevent the biofouling of materials. So many groups, and our group, have demonstrated that pathogenic bacteria such as Candida albicans, fungi such as Candida albicans, and pathogenic bacteria such as Staph and Pseudomonas, in the presence of these slippery surfaces, they are not able to form biofilms on the surface, and they just slide off. The same happened for the bacteria as well. But what we were more interested in, and we were more concerned, is that it's happening to those bacteria that are in solution. So for many applications, this will be great. It will be fine to have a surface that is anti-fouling. But for applications, for example, in the healthcare or for biomedical equipment, you want to develop surfaces that also affect or kill the bacteria and prevents the anti-fouling. Because we can begin thinking that this may cause infections to patients, and then it can become very problematic. So how can we turn these surfaces that are anti-fouling inherently to make them to become antimicrobial? So the approach that we use to design to make these surfaces to become antimicrobial as well, it was that if we can hold liquids inside these surfaces, then we may be able to hold other molecules inside of them. Molecules that are active and compatible with the polymer matrix that we're using, and that can then kill or affect the behaviors of the bacteria such that we can control the virulence or the pathogenicity of these bacteria. And we have demonstrated that by loading a model broad-spectrum antibiotic, triclosan, inside of these films. And we have seen that by loading triclosan into these films and adding the oil, the slippery surfaces remain slippery after adding the oil. And furthermore, when we expose these surfaces to aqueous media at a physiological environment, we can then start to slowly releasing the triclosan that was loaded inside these films over a period of more than 4 months, and that the amount of triclosan that was being released onto these, of these surfaces was at relevant concentrations that was able to do 2 things. One, that it increased the efficiency of the sleeps by reducing the viability of the cells that were on the surface of the sleep, but also reduce the viability of the cells that were in solution significantly when we compare to the bare surface and the sleep as well. But our research group is also interested in how can we not both use biocidal treatments, but also how can we use other type of treatments that will prevent the bacteria to become pathogenic without killing them and potentially preventing them from becoming resistant to these type of treatments and these type of surfaces, since we will be releasing these agents for longer periods of time. So quorum sensing is a new approach that can be used for that. Quorum sensing is just a basic mechanism that bacteria uses to communicate and then engage in group behaviors, such as the formation of biofilms or the production of many different virulence factors. So our collaborators here at UW-Madison, Professor Helen Blackwell, she's an expert of designing molecules that can inhibit these quorum sensing mechanisms of bacteria. So in their group, they have developed a series of different quorum sensing inhibitors, and we wanted to see whether we can load these quorum sensing inhibitors in our surface and if we can prevent the virulence production of these bacteria by measuring the production of piocyanin from the bacteria that was in solution. So after loading these quorum sensing inhibitors, we saw that depending on the structure of the quorum sensing inhibitor, we can control the amount of quorum sensing inhibitor that was being released, and that will, depending on the application that you want to use, you may select the quorum sensing inhibitor that is desired. And furthermore, we observed that the quorum sensing molecules alone did inhibit the production of piocyanin, this virulence factor from Pseudomonas, but furthermore, the approach that we're using allows us to combine different antibiotics and different molecules into these surfaces. So we saw the possibility of incorporating 2 of these quorum sensing inhibitors and see how these prevented the production of this virulence factor. And we saw that it greatly reduced the amount of piocyanin that was being produced. So we have demonstrated that we can employ these surfaces and these interfaces to create materials that are both antifouling and antimicrobial, and that could be beneficial for a large range of applications in the biomedical field. And we have different tools and targets that we can tailor to make these surfaces as specific for any application that we want. So with that being said, I would like to conclude this presentation and give thanks to our collaborators and the group members that make all of these possible, as well as the funding agencies. And if anyone has any questions, I will be happy to answer them.
Questions for Benjamin? I have a question for you. So the quorum sensing inhibitors, I'm wondering, how do they work? Do they have to be taken up into the cells to exhibit their activity? And what I'm wondering is whether one could think about immobilizing quorum sensing inhibitors on surfaces, because if they don't have to be taken up into the cells, then they may be active when immobilized. Do you know how they act?
So usually the quorum sensing inhibitors, they will bind to the cognate receptors that cells have, to the receptors that cells have. So usually they will have to internalize inside the cells, and then yes, they will need to internalize and then bind to these receptors that cells have to initiate their transcription. So if it doesn't bind to the receptor, it binds the complex that it forms inside the cell. It's a complex that it's not active. So it prevents the transcription of more quorum sensing agents and prevent the transcription for the production of all these virulence factors that bacteria produces.
So are there strategies to interrupt quorum sensing which are based on recruitment of the quorum sensing molecules? So depleting their concentration? Because that would be another way of inhibiting communication between cells.
Yes, that's something that we have been thinking about. We will have to look at the structure of these quorum sensing molecules and then design other molecules such as cyclodextrin or other molecules that can recruit those quorum sensing molecules that are being released by bacteria such that we will have a very low concentration. But yes, it's a really good idea to prevent quorum sensing activation.